So firstly, thank you ever so much for Jeremy and Promian for inviting me to speak. So yes, very much at home in Oxford. Real pleasure to be back here. I see with the conference we've got a range of immune responses that we want and we don't want. I'm definitely in that camp of this is the unwanted immune responses. I've already had a couple of questions about it, so I will preface this talk by saying at Pfizer, we recently had a phase 3 failure, which was put down to immunogenicity. So we have a lot of interest in it. I obviously am an ambassador for Pfizer, so it is our responsibility to do things differently. But that molecule was developed a long time ago, and we didn't actually apply these kind of tools. So got my little get-out clause as well. Okay. Okay.
That's the next one.
This technology is so tricky. The down arrow on the computer works. I'll stick with that one. Okay. So for immunogenicity in a negative sense, it pretty much affects every biologic that's developed. So I don't think there's any biologic that is completely immune response free. And there's quite a lot of burden that goes along with that, even if the immune response is somewhat benign. But really, I guess from my perspective, I kind of assess the risk for all of our programs as they start very early on preclinically, as we start to get data, and then through the clinical space as well. And the bits that we're really interested in, the message I have to give within Pfizer is one of immunogenicity can have a big impact. This unwanted response certainly does lead to a loss of efficacy. From the company perspective, a loss of revenue. Obviously, from the patient's perspective, having to come off a drug is a big deal. And just kind of highlighted here, if I've got the technology right, this is actually patients who were taking these TNF inhibitors, 3 well-known, big grossing drugs. It's known for rheumatoid arthritis that the first 6 months of treatment, lots of people come off the drug because it's just not working for them. But then there's this kind of loss in patients. So this patient's still on the drug from 6 months. It's actually a Danish study. I think there's about 8,000 people in this. And the rate at which people were coming off the drugs, it was kind of usually that there was either an adverse event or there was a loss of efficacy. It seems that probably about half of that is due to an unwanted immune response. So adverse events kind of such as hypersensitivity can occur once you develop the immune response, or the drug can get cleared more rapidly by the anti-drug antibodies. So that's kind of, I guess the thing to point out sometimes, like Humira, which I know Jeremy has data on. I'm sorry, I don't know if you presented any of that today, but Humira, which is the biggest selling drug, actually has some liabilities, and we know that there's a quite high degree of immune responses that occur. And so, there's a lot of patients who are having to switch. And that's kind of fine in rheumatoid arthritis. You've got a number of other options, and not just the ones shown here. In other diseases where there might only be one treatment, or certainly where we're trying to make innovative therapies where there will be only one treatment, well, yeah, this isn't really an acceptable position. You give someone a lifeline, or at least a way to mitigate their disease and to feel better every day, and then within 18 months, 2 years, it's not working anymore. That's clearly not a very good proposition for the patients. I'm pretty sure the NHS wouldn't be happy with that either. Just to highlight as well, there was another kind of more significant failure in a sense, is the likes of, I can't even see the thing there. Okay. Novo Nordisk are actually working on a coagulation factor, so a factor 7A, and essentially they tweaked it a bit to give it a better half-life. And that actually introduced T-cell epitopes. So the patients who were receiving that drug, if they had the right, or in some cases the wrong HLA type, and then they'd been dosed for a fairly long period with that drug, they began to get the anti-drug antibodies. And in 2 cases, they were cross-reactive with endogenous factor 7. A, and that can lead to the potential that the patients won't have any clotting system left. So this is serious enough to not continue development of programs like that. And again, this is like phase 3 by the time you find this out. So that's a lot of money you've wasted in a sense, when you probably could have made an assessment with an in silico package with some nice in vitro assays to actually get you to a point where you realise you've got a liability that you need to change earlier on. So really the key about understanding the whole immunogenicity process is that you can plan and develop safer and more effective biologics, and certainly you don't have any embarrassing phase 3 failures. So just kind of questions around the sort of approach we take for immunogenicity strategy. Appreciate there's sometimes a bit of a difference between a biotech approach where maybe there's a couple of drugs and you want to kind of throw everything at it, make sure we've definitely got a really good product. And then a company like ours, where I think we've got like 90 biologics on the go at the minute, and we have to be a bit selective about how we're applying the resources. So I would also second the in silico approach as being very favourable. But really, when we've observed immunogenicity, that kind of brings with it a big regulatory burden, that the regulators are very concerned as to, well, what will that become? So we end up having to do more assays, or potentially set criteria by which we wouldn't continue development. Just to kind of highlight, I guess, this new environment, there's been various guidances out, be they for monoclonal antibodies, be they for biosimilars or other protein therapeutics in recent years. And this really does kind of drive us to say, it's not really satisfactory to just sit back and see what happens, but actually to be more proactively investigating the biologics in development. So I was at a meeting towards the end of last year, and we had work for an American company, right? So we're very FDA focused. If the FDA says something, we have to respond to it. So I do like to show the slide where we have the FDA reviewer saying, well, it's going to happen, right? Immunogenicity will happen, and we need to do that analysis early on in development. So it's still not necessary to have done all of the in vitro assays and the in silico prior to first in human, but it still is seen as a positive from the regulatory agencies if you've done that due diligence, and ideally if you can show that you've got low responses in those assays, of course, very helpful. And we do as well say within Pfizer, because of the number of assets we've got, we do focus our resource on those higher risk assets, and indeed all of our most of our attention on reducing risk at the higher end as well. So there's quite a lot of information on this slide, just really to kind of pick up on how the different risk factors for immunogenicity sort of fall into a few different buckets. So those related to the product. We heard earlier about the assessment, it doesn't necessarily just stick with the monoclonal antibody or protein that you're actually trying to get to do the job, but also potentially some of the impurities or degradants that you might have to consider as well. Patient factors are very important, often very early on. We have an idea of who the patients are going to be, and we can't really do much to change that. And treatment as well, sometimes I think that particularly for long-term chronic therapies, that we're just going to have to live with the fact that there's a chance that people will generate an immune response, like with the TNF inhibitors that I showed previously. But these are kind of all of the different areas we're thinking about when we're doing risk assessments for our products. I guess you've probably seen this kind of cartoon of the immune system already. Pretty much all of us have a model of the immune system in our head, and this is the one I kind of spit out onto paper, or at least onto the screen. And kind of historically as well, we kind of went from, we put the therapeutic protein in and we measure these anti-drug antibodies that are coming out the other end. But we know quite a lot about what goes on in the immune system. It's almost like another month doesn't go by with a new subset of T cells, but essentially we've got the bones of the immune system here, and yes, there are differences between us, but it's generally operating in the same way. And this was really, I guess, a big philosophical move for us to say, So the immune system is different. It is hard to predict, but actually there are some underlying commonalities. So let's focus on those. As you've probably already heard about, the antigen-presenting cells obviously key in taking up protein. We find, as do others, if you've got your protein, this is a bit of a horrible glob really, but say it was a monoclonal antibody that was targeting a receptor on an antigen-presenting cell, well, that's a great way to deliver that into the antigen-presenting cell for it to be processed and then presented here, so on the HLA. We also find as well that we've got some remarkably sticky antibodies. I think there's sometimes discussion about whether you should use an antibody that's from a mouse that you graft onto a human, then humanize, whether you should use a phage display, because that's a great way to find antibodies quickly. There's I don't know how to put this, maybe it's a belief, a bias for some people that if you've got a phage display antibody, it's not gone through the same kind of selection process. So it's more likely just to be sticky, and hence you might get this sort of more non-specific interaction that then leads to uptake. So this is something we're actively investigating at present. The other pieces here, obviously the T cell will both interact with the antigen-presenting cells and the B cells, and ultimately that's around offering the help to the B cells to lead to this antibody production. I guess as well from my perspective, and probably some of the other assays you've seen, this is a really interesting part. A lot to do with the fact we've got the tools and we can investigate it, and we believe we've got a process by which we could essentially remove these epitopes and therefore pretty much cut the T cell out of the picture. So that it has been a suggestion that if you got rid of all of these T cell epitopes, although you still might see ADA, that they would generally be of lower affinity, perhaps you wouldn't get class switching as well, and so you'd be able to continue dosing your patients without this kind of issue. So I'm happy now as a technologist rather than a scientist, thinking about, well, what's the practical approach we can take to make sure we can keep patients on a drug, or indeed actually get the drug to the market so the patients can see the drug. At present, this is very much our focal area. Just to kind of reflect a bit as well on sort of the process by which we try and make improvements. And this is kind of a bit of a process map, right? So I talked about being pragmatic. We need to make some choices. We need some data, and we need to work out, well, what's our likelihood of success? So we know the problem is the unwanted immune response. There's probably a range of different solutions that we could apply, and some of them were just not going to be useful, right? When I first started in this field, which is probably only, I guess, 7 years ago, the position was from most people, you can't predict immunogenicity. And that's kind of fine, but it sounds like a challenge to me. So I go, yeah, okay, let's see if we can. I guess this confidence level is key as well. Again, as a scientist, we want 95% confidence, right? At least. In business, we often settle for an 80-20, although I would say some of the decision making I've seen is probably 50-50 is good enough. And certainly then when you ask people, well, if I'm going to predict immunogenicity, what level of confidence should we have? And some people there are happy with 50-50 because it's way better than they thought they could have. I'm kind of still open to saying, well, let's make some predictions and see how well they turn out before I tell you where we are on this. So that's not the punchline. You won't get that information today. But really just to say, well, let's look at some different solutions and apply to clinical outcomes and then go through some refinement. And at some point, implement a great solution. So this was kind of the solution one. And I appreciate we've heard a bit about in silico tools, some of the T-cell epitope tools and in vitro assays, and then the animal models, including up to like humanized mice. It's really tricky to look at all of the different risk factors. I know I glossed over that slide with the 3 buckets, but if you look at guidance for EMA or FDA, there's like 13, 14 different factors that you need to take account of. So how do you integrate the information that you generate, interpret that, and then actually try and correlate that prediction with some kind of outcome? It's kind of I guess, not so feasible just by running a few assays. Sure, the assays are asking specific questions, like is there something in my sequence that looks like it could bind to an MHC molecule? And that's important information if you want to change the sequence, but it doesn't tell you what clinical immunogenicity is going to look like. So a few years ago, whilst we still had a view of what does this look like for the wanted side of the immune response, when we were working on vaccines, we've kind of played around with bolting together a few models. Now, this is complex, so you don't necessarily need to look at all of the pieces, but just give you an idea of the sort of structure we work on. That when we're building a model, we basically have got different variables and then fluxes between the variables. So there's kind of B cell numbers, T cell numbers. And again, it's not really to say this is our model we have. This was an attempt to bolt together some models that were already out there in the literature. And I always love the kind of looking back on some of these and going, right, well, a lot of our B cell response in this model was taken from a 1970 paper. So that was a little bit before I was born, but nevertheless, it's still very good science and a great starting point. Yeah, you certainly shouldn't reject something just because it's old, but what's the context? How can you put it together? And although we kind of bolted these things together and showed that we could reproduce some immune responses, and this is all in silico, right, inside a computer. You don't even have to worry about going to a lab. You can just sit there and play games. So this kind of gave us we can predict an immune response. You know, kind of, it's easy to predict with hindsight if you know what you're trying to predict, right? So there was a few more challenges along the way. At this point, there was enough interest in Pfizer that we were obviously dedicating our massive resource. We put one postdoc on a program. And she went, well, it's kind of interesting, but it's a bit complicated. I'm not sure we need all that stuff. So she kind of tore down everything, went back to the literature, and basically kind of said, well, what's the prior data we're interested in? You know, what kind of bits, the epitopes, the in vitro, with bit binding or proliferation, and then aspects of the system as well, so populations, genes, any kind of co-medication people are on. And really just to try and put all this information together. So essentially build an immune system within the confines of the computer. I think there was something like 140 variables that have gone into the model. Sometimes we look at that and go, well, that's a big model, that's tricky to process. And then some of the other people we're working with were going, right, well, if we're looking at drug-induced liver injury, we've got like 500 different parameters there. So, you know, we can squeeze a few more things in this and we won't be stressing our processes too much. But really the outputs that we're looking for as well. So the clinical ADA incidence is an interesting one. So this is often a number that people will band about, and indeed so much that it's actually on the label of the biologic drugs. It's like how many people in a phase 3 study generated an anti-drug antibody response. I think what's more meaningful is the loss of efficacy. So if you can predict, like maybe for the TNF inhibitors, that you're going to lose 50% of people over a 6-year period, now build that into your thinking about, do I start earlier to actually make a better molecule? We're kind of intrigued to think about optimal trial design. We've not yet applied that. When we were looking from a vaccine perspective, we found some interesting things out there which we were hoping to try and never quite got around to. But as well, the bit that kind of sometimes takes me back when I'm sort of daydreaming about being a scientist again is actually to understand some mechanisms and actually use the model to ask questions which can then inform experiments that we can do differently. Okay, so again, probably kind of a simpler view of the model, if you like, that is built on these multiple levels. So obviously there's immune cells that are involved, and that these take place in the context of tissues and organs. And for an individual, we've got all the whole body level of, well, you know, what's the inputs into the system in the way of the drug, what's the anti-drug antibodies that are being produced, and then what's the elimination of the drug because of those ADA. And then the population level as well. And clearly the MHC class 2 genotype's important in this respect. And we certainly believe that some of the other couple of thousand immune response genes may well also play a role here. So the initial approach is to try and predict the outcomes for a population, where in future we would hope to refine on the basis of actually having data from individual subjects, so that we can kind of see what are the most important factors and how do we incorporate those in the model. Okay, so I'm just going to skip over, because one of the key things when we do build this kind of mechanistic model is to understand the sensitivity. So there's a few key things here, and Bernard likes this slide because we've got the naive T cell number up at front here. And you can kind of see that from some of Bernard's data, hopefully I've got this bar in the right place. So this would be the usual range by which we would see the number of naive T cells in across the population for biotherapeutics. And what we see here is, sorry, this scale looks a bit mashed, my apologies. But essentially this is an increasing amount of immune response based on the number of naive T cells that are there. I don't worry too much about the right hand side of these graphs. There may be something to do with tolerance going on. We may not have modelled it exactly right, but in essence, to highlight really that the naive T cell number across this range makes a bigger difference than the naive B cell number. That was nice when the postdoc showed me that, because I'm very biased and think that, well, the T cells are going to be much more important, and we've got some assays to actually look at that, so that's really helpful as well. Again, probably no surprise when you look at the epitope MHC interaction, it pretty much falls off a cliff here. And we can find potential epitopes of all sorts of different strengths. I know kind of from the talk earlier there was an EpiVax approach, and they're pretty good at saying you need to go on and test these. But I think there was probably they would kind of capture things that go a bit higher than we would capture in the context of this model. One thing that's a bit annoying actually is this antigen dose or drug dose. So for most biotherapeutics, we were kind of finding that they're dosed in a range that would give kind of the maximum ADA response. So, there are some ways to get around that, right? Great potency, fantastic half-life, that kind of stuff. So of course we're working on those as well, but for now we're a bit stuck with this being in an awkward position. So I kind of tried to summarize all this. We actually published 2 papers kind of back to back in 2014. One of them's about kind of all that mathematical stuff about building the model, and then the other one's the application of the model. But to kind of highlight here that we've got our antigen presenting cell, so we've got our drug and other antigens in the system being taken up, some degree of competition for the peptides that get presented. So we kind of understand this comes from like the protein. Obviously there's the host MHC types key in there as well. And then the host specific parameters around drug clearance as well. As I said, we don't just want to say, did you have ADA? We want to say, did those ADA actually cause you to have an enhanced loss of the drug and therefore a loss of efficacy? So when you kind of do this model simulation, you can go for an individual. In fact, we did for 1,000 individuals, and we were able to demonstrate this is epitope MHC combinations. And in adalimumab, we know there are 2 sequences that are well presented that are of non-human germline. So we basically said these could be the 2 epitopes. Those 2 epitopes are only predicted to bind to DRB, not DP or DQ. So we kind of said essentially, as an individual subject, you could have 2 epitopes and 2 of the MHC that present. So a maximum of 4 epitope MHC combinations. And actually, whether you've got 2, 3 or 4, it kind of left you in the same bucket here. That's enough of a stimulus on the immune system to give you a kind of maximal response, at least as far as our model was concerned. And then if you didn't have any of these epitopes, well, essentially you're kind of bumping along the bottom. There may be some sort of, I guess, natural antibody, if you like, potentially just the IgM response occurring. And so what we found significant then was that it would only be these subjects who would likely see a loss of efficacy because you're binding up drug, making the complexes and leading to the clearance. And this simulation, again, sorry, it doesn't look like this is too clear, but this was best part of 600 days. And that was in part optimal for the running of the program as well. So it's a little bit of an odd time frame. But what we were trying to show here was this drug survival concept. So if you were in a position where you would see a 50% reduction in exposure leading to a loss of efficacy, you'd see people losing efficacy pretty quickly. So it may be that it's more realistic to think that you're in line with one of these other enhanced clearance rates. The other thing that we've included here is just one standard clearance rate for a subject who has ADA, and we suspect we need to get more data on that to really kind of understand the individual response better. Okay, so just highlight another part of the kind of analysis you can do with this model. At the time we were discussing with people who were working on biosimilars, well, what happens if we got, a 50% ADA rate and someone else gets an 80% ADA rate? Is that significant? And actually with the typical trial size for biosimilars, you're more in the 50 to 100 range. So our projection with 1,000 subjects is the blue line, essentially, that you can see that in our projection we would get 75 to 80% immune response to adalimumab. Whereas if you were to take 50 subjects at random from that 1,000, they could plot out these different circles. So these were, I think there was something like 100 different simulations done. So this is the range you could get, which was comforting to our biosimilars team, who had found actually that our own drug was giving less immunogenicity than the comparator that we were trying against. And also at about the same time we were seeing data come out from Amgen and I think it was Samsung Biochemistry. One that was more at the 50% rate and another was at the 90% rate. So, it's kind of, in a sense, this is part of the witchcraft bit, right? It's like, well, it could be anything. But in a sense as well, it does kind of help you understand that this anything is predicted on the basis of if you don't know what your HLA types are, and this was North American population. But if you don't know what your HLA types are, it is going to be difficult to predict. And not because of this, but for other reasons, we are now doing the genotyping to get the HLA type in all of our clinical studies. So hopefully we're going to get a better understanding of how accurate some of this approach is. I guess there's a number of different questions we want to ask at different times. And I think kind of starting the program and trying to run that whole in silico prediction about what's going to happen in phase 3 or on the market, isn't really going to carry much weight with the protein engineering and the early discovery teams. So, we kind of want to ask some questions, and appreciate as well we heard on the Affibody one. Well, sometimes we just want to say, well, which is the best candidate? So which one's got the highest risk? What should we take forward? Ideally, in a sense, it's like this piece, right? Influence the engineering. So you're kind of not looking back and going, oh yeah, so we took the best one forward, but we still had a problem. But actually we did something about that T-cell epitope risk because that's the thing that we could control. There's a few pieces here about kind of the non-clinical development. We've not applied this to non-clinical studies, although we do have non-clinical versions available. But sometimes we get this ADA limiting the interpretation of the study, be it from a tox perspective, lack of exposure. Sometimes we'll get findings that no one can quite explain. I'm not saying that our model will solve that right away, but it certainly can be helpful sometimes in interpreting what you see in a preclinical species compared with what you're going to see in the clinical trial. I guess there's questions for later clinical development as well about manufacturing changes. What we don't build in as yet are things like the changes in impurities. I was hearing recently that Genentech had to go back to a phase 2 study because the material in their phase 3 had a contaminant that was very immunogenic. All of those aspects of risk you do need to address as you're going through. just kind of highlight as well, actually, in the early clinical development, suitable bioanalytics in place so that we know that there's tolerance to the drug and also any target. And actually this was one of our problems for the bococizumab trial, that we were kind of using old assays for phase 2, and then we kind of looked at using a new assay and looked at some incurred samples and kind of saw a higher immunogenicity rate than we were expecting. So it's definitely good to keep up with the latest technologies, which you'd hope to do as a biotechnology company or a wannabe biotechnology company. So this is a slightly different angle. So I've talked about T cells and antigen presentation. There is, of course, the B cell response, which I sometimes forget to mention. But one of the approaches we're looking at here is actually to try and use a germlining technique. I guess we tried to get it called ultra-humanization, but PNAS didn't like that, so we had to settle for augmented binary substitution. A bit more of a mouthful, but that's you can take away ultra-humanization, I don't mind, that's fine. And really the approach here is to say, well, if you kind of start with a molecule, you're making a graft or a parental molecule from a mouse, and you graft it onto a human. Essentially all of the coloured residues are the potential bad guys, right? They don't look like the germline, so you could well generate an antibody response to those. And here as well we're showing the kind of binding to the target. So when you go from the parental to the graft and then to one that's gone through the ABS process, you're basically reducing the amount of colour on the surface. Inevitably you've still got, it's actually the CDR of the heavy chain, CDR3 of the heavy chain, which isn't encoded in germline, so that's always going to be different. So you can't get to 0 epitopes, but you can at least get to reducing your risk level. And these are interesting. As far as the paper was concerned, the T-cell epitope piece was a bit of an afterthought. So we actually used the EpiVax algorithm here, and we were able to demonstrate that if going from parental to graft to then the ABS versions, we didn't necessarily get rid of all of the epitopes. So this has served as a good reminder to the protein engineers to think about T-cell epitopes as well as they go along. And indeed, which framework are you putting your CDRs into to make sure you end up with a reduced immunogenicity profile. So relatively early days, but we have had a couple of these recently where not only do they look sort of like this and maybe even tighter, but also we've got no predicted epitopes. And we did recently use Proimmune's peptide assay to demonstrate that we did also see no T-cell epitopes. So even though the in silico hadn't called them out, we just went, oh, let's just test this anyway and make sure, because this is something we want to use more frequently now as well. Okay, so I kind of talked about some different solutions. We're kind of doing some assays and kind of waving your hands a bit and guessing, to doing a computer model and still waving your hands about and guessing a bit, to actually getting to a point where you can more quantitatively predict with some degree of accuracy. I think we should have a discussion over what degree of accuracy we should be aiming for, but I'm very happy to have that. But really kind of going from solutions, what's our next solutions? We've got a framework to work on. We've got a number of molecules that are going through clinical development right now that we're applying these to. So the in vitro assays to inform the mechanistic modelling approach. So we need to get more preclinical data, more clinical data. If you talk to statisticians, all they want is more data. They're never satisfied. And we know there's room for improvement in the model as well, be it through risk factors or immunological factors. Most importantly, it's applying the model, right? So this piece, let's apply it prospectively. I want to stick my neck out and say, this is what I think your immunogenicity in your phase 2 is going to look like. And then hopefully we'll be right enough that that will say we're in a good place to carry on and take this to a phase 3. So just quite a lot of people have worked on this. I'm going to highlight at the top here Xiaoying Chen, who my postdoc, who when she was first producing the model said, I found loads of great data. It's about over in mice. I went, oh no, not more mouse work that's not going to be translatable and causes pain. But she did it. It helped her learn a lot about immunology and helped to build the foundation of the model that we've got there. And then formally with Pfizer, Paolo Vicini, so modelling guru who knew nothing about immunology, but we had some great discussions, and between us we kind of cobbled together a model. And we've worked with a number of external partners as well. So thank you very much for your attention. Happy to take questions.